Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience from Harvard University, where I focused on neuroendocrinology and the neuroscience of behavior. And before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to parse and translate the information guests share on the show. And in addition to the podcast, I write long-form content and send out a free weekly newsletter at the Mind and Matter Substack at mindandmatter.substack.com. I also offer private, science-based health consulting, where we work one-on-one and apply the same rigorous, evidence-first approach you hear on the podcast, but personalized. We can obtain and analyze your biomarkers, work on diet and behavior strategies, review supplements and drugs that you might be taking, or anything else related to general metabolic health. So if you'd like more tailored guidance on your personal health, head to nickjikomes.com and fill out a short form or book time with me there. A great way to support my content in all of its forms is simply to visit mindandmatter.substack.com. You'll find a link to that in the episode description, and you'll also find a link to my current affiliate partners through my support page. These are products that I use myself that relate to physical, metabolic, or mental health. For example, one product I use every day is the AquaTrue water filter, which removes heavy metals, microbes, endocrine disruptors, you name it, so that you have higher quality drinking water. All of those links for health consulting, for my affiliate partners, and the Substack page are all in the episode description. So thank you very much for listening, and don't forget to check out mindandmatter.substack.com for all of my content. David Lee, thank you very much for joining me today.
Thank you for having me here, Nick. It's a pleasure to be on your podcast.
Do you want to start off by just telling everyone a little bit about who you are, what your background is, and what your lab studies?
Yeah, my name is David Lee, Changhan David Lee. I'm an associate professor at the University of Southern California, Leonard Davis School of Gerontology. And as the name of our school implies, my main focus is studying the biology of aging. And we focus on mitochondria in our lab. More specifically, as I'll talk about more later, my lab is very interested in understanding how mitochondria communicate to regulate things, in addition to being the so-called powerhouse of the cell, as well as what I call the slaughterhouse of the cell. We think mitochondria are what I call the speaker of the house, that regulates things that are relevant to aging.
So the slaughterhouse of the cell, what exactly does that mean?
So it's a play on words: powerhouse, slaughterhouse, speaker of the house. Powerhouse, you probably know, generates energy. It provides building blocks for life. And the slaughterhouse is because mitochondria are very integral to what's called programmed cell death. So when a cell deems that a certain damage is too much and it's beyond repair, then it activates, it's called apoptosis, which is a programmed cell death process. It's also important for development and other processes like your digits, your fingers. The way they're formed is through these programmed cell death to separate out certain structures. And therefore, mitochondria being one of the most important regulators of this process is, I call it a slaughterhouse. And the speaker of the house, obviously, because it talks stuff.
So mitochondria are at the epicenter of bioenergetics. They're making most of the ATP for our cells. Obviously, that energy is used for building things and making things, but it's also going to be an important part of tearing things down, cell death and things that you were just talking about. And whether we're talking about building things up or tearing them down, ATP has to be used, energy has to go places and fuel different cellular processes. And the mitochondria, of which there are usually many inside of our cells, have to talk to other parts of the cell, so to speak. So you've been involved and I think discovered some of the amazing communication molecules that we're going to talk about today. But give us, at a very general level, a basic rundown of how does something, an organelle like mitochondria, talk to other parts of the cell? What does that look like at a basic level?
So there, excuse me. So I kind of think about it out as a mitochondrial language, of which the alphabets are many. And there are several that have been pretty well defined. One of the more fundamental ones are the metabolites themselves, such as aspects of the electron transport chain, like ATP, NAD, you might have heard of. These things can communicate, act as a communication factor through epigenetics, which means it can influence how the nuclear genome regulates or operates. And then there are other proteins that are encoded in the nuclear genome that can live in the mitochondria, and they go back to the nucleus to report back on certain situations. Mitochondria, I'll touch upon a bit later more, being derived from bacteria, still retain a lot of these bacterial components. And the leakage or the export of some of these more bacterial-like compounds or molecules can stimulate the cells to recognize it as an immune response or inflammatory response. So there are many ways to do that. Mitochondrial DNA itself is considered an inflammatory signal, a part of our immune system. But then what we study in our lab are little proteins that are called microproteins or peptides, depending on who you talk to. They're encoded in the mitochondrial DNA and are used as communication factors. So they're very versatile in the way they communicate.
Yeah. So we've got a nuclear genome. The genome that we normally refer to is the one that's in our nucleus. It contains most of our genes. But we've also got these mitochondria, which used to be free-living bacteria that became incorporated into larger cells. They have their own mitochondrial genome, their own mitochondrial DNA that encodes certain things. And there has to be communication between both genomes because there's actually things encoded in the nuclear genome that are part of the mitochondria. And there are things, as we'll learn, encoded by the mitochondrial genome that can go into the nucleus or talk to other parts of our cells. So there's a bidirectional communication at the level of genes and gene products between the mitochondria and the nucleus and even other parts of the cell.
Yeah, no, you put it really well. That's actually a very important aspect of this communication. Maybe you can go back to the very beginning of this relationship, perhaps. So the way we like to think about it in our lab is going back to the origin of eukaryotes. So rewind 1 billion to 2 billion years, depending on who you talk to. Before we had eukaryotes, there was our ancestral cell, a bigger cell, and then this really special bacteria that was good at extracting energy. And the bacteria, in a way, infects the bigger cell. It invades it and enters the bigger cell. Some people call it a marriage, but it's basically an infection. And it decides to live in there, right? So typically when an infection happens, one kills the other and one reigns supreme. But in this case, something weird happened where they figured out that not only is it beneficial to live together, it will be good enough to make it permanent. So the bacteria became a mitochondria, right? That's called the endosymbiosis theory that was first posited by Margulis. But what we think was really important here is the communication started from there. And that was the most important thing, because to coordinate something as complex as integration of two independent life forms, basically two sovereign countries coming together, must have had a lot of coordination required, which absolutely requires communication. So how they communicated, how they talk to each other, I think is where things started. And they probably didn't invent a whole new language to start to talk to each other. It's an infection, right? So a lot of these would be tooth and nail kind of situation. A lot of these supposedly immune factors that they use to control other organisms may have been used. And there are those today in bacteria. Those are called antimicrobial peptides. Basically, every living form or kingdom on this planet has these very ancient immune peptides. We have it, flies have it, birds have it, bacteria have it, plants have it. Everybody has it. And they are used as the next generation antibiotics commercially. It's one of the frontline development for antibiotics. But we think those peptides might have been used to talk to each other, to control instead of kill, because a lot of these peptides have very specific functions. So the first line of attack would be bombarding the membrane and disrupting the membrane using its characteristics. It has to have a positive charge and it has to have a hydrophobic domain. And they use that to target the membranes. Once it gets in, many of them have intracellular targets that are vital. These are evolutionary selected ones. So they don't fool around with minor processes. They go for the jugular. So DNA synthesis, RNA synthesis, ribosomal function, cell wall synthesis. And these are very well defined today in the ones that we know today. But if you think about it, the bacteria's biggest fear would be dilution, because in a way, it's paying rent inside this bigger cell and has this benefits in living inside it. But if the bigger cell decides to divide without them, if the division is not synchronized, after a couple of divisions, they'll be diluted out.
Right,
So the
right.
theory that we have, yes, so the theory that we have is that somehow the bacteria had to have a way to control the cell division so they're synchronized. And one way they could have done it is by using these, I guess, hostile communication factors, typically, to dial it down. So instead of bombarding it with high concentration, if you dial it down, DNA synthesis will be halted, but it won't be halted forever to cause cell death. It'll just slow down the division rate. So if you fast forward that another billion years, today we think this is a foundation of cell regulation. So when a cell starves, how does a cell know how to regulate its metabolism and slow down cell division? We think that is the foundation of how this so-called cell regulation established.
Yeah. So even pathogens, bacteria, viruses, our cells have to communicate with them and respond to them, and that should make kind of intuitive sense to people. Like even if we get infected with a bacteria, that bacteria is going to have an interest in shutting down our immune responses, modulating the inflammatory signals that our cells produce. Obviously, our cells are going to have an interest in getting around that, at preventing our own cell machinery from being hijacked. There's going to be crosstalk there. And so a lot of that crosstalk will naturally involve things that we think of as immune functions. And so if we sort of think back to the endosymbiotic event by which bacteria that became mitochondria got permanent residence within our cells and eukaryotic cells, they had to modify this language or modify the communication so that they could cohabitate and they could play nice, so that our immune reaction wouldn't be destroying them, so that as you said, the cell cycle stuff was synchronized so that we retained mitochondria with every cell division, and then obviously so that the bioenergetic stuff could be incorporated into the cell and put to good use. So it sort of makes good intuitive sense. There's going to naturally be communication between these entities, the nuclear genome, the mitochondrial genome, that it's going to involve things like inflammation and cytokines and stuff like that, and so on and so forth. What I want to talk to you about specifically are these microproteins or peptides. So let's start at a basic level before diving into MOTS-c, which will be our focus today. What are peptides? And can you give people just a general sense for, are these very numerous? Are they everywhere? Is there a whole bunch of them? Are they highly specialized, weird mitochondrial things? What are peptides, and what does the landscape generally look like?
So peptides, at a very high level definition in my mind, is just small proteins. But I guess the debate comes down to how small is small. And if you talk to a lot of academics, I guess right now there's a broad consensus of roughly about 100 amino acids being the cutoff to qualify as a peptide. But of course, there are ones that are slightly bigger and very, very small. Like the ones that we study in our lab that we named MOTS-c is only 16 amino acids. If you ask FDA, they define it as 40 amino acids and less. So 39 and below are considered peptides in the eyes of the FDA. So there really is no universal definition of what a peptide is. But there are many sources of these smaller proteins. They can be processed, smaller peptides that are processed from a bigger mother protein, like insulin, for example, is considered a peptide hormone, but the proinsulin is much bigger. But then there are the fragmented ones too, those that we're not sure if they're all processed intentionally or if they just broke off. They're the smaller fragments of the bigger proteins or peptides. And then there are ones like the ones that we study that are encoded in a small gene. The gene itself is very small because it's intended to encode for a small protein peptide. And many people now use the word microprotein to kind of uh, differentiate between synthetic, artificial, and broken pieces of proteins that are small peptides, and then ones that are gene encoded and ribosomally translated intentionally to be small. So different words.
Yeah. And now when we think about natural endogenous peptides, are these things always explicitly encoded? So like a small gene encoding a small sequence of amino acids, or can they also result from like the breakdown of larger proteins?
Right, right. So like I just said, like insulin, for example, it's processed form of a bigger protein, proinsulin. So that's already very well studied. The smaller genes have recently emerged, and for a long time we didn't know of the existence of these gene encoded small gene encoded peptides or microproteins, and that kind of goes back to the Human Genome Project, in the 2000s, late 20th century, when we sequenced most of the human genome. We thought we had the map, and the string of ATCGs came out, and to map it, you have to annotate the genes, right? And during the process of annotation, there were many criteria that were involved, but for various reasons, these smaller genes were not included in the annotation. And that roughly kind of cuts off around 100 amino acids for many reasons. Go into detail if you want to, but for that reason, if you go and run all the proteins in a human sample, for example, using a mass spec, you would see the existence of these smaller peptides, but there was no gene to ping them back to.
Yeah.
So there was no way to identify them. But now with increased technology and the advent of all these, informatics and tools that we have now, we're starting to see these little genes and the little peptides that they make. So there's a floodgate that's opened up in the nuclear genome. Many have been discovered, as well as the mitochondrial genome.
Got it. So there's a lot of peptides out there. By definition, they're small proteins. And for historical and technological reasons to do with things like the resolution of our methods, these things have always been there. They've always been abundant and probably doing important things. But to some extent, we sort of missed them or couldn't quite detect them until relatively recently.
Exactly. Yeah.
And so we're going to spend most of the time today talking about a particular mitochondrial peptide called MOTS-c. It's getting a lot of attention. It's really interesting. Talk to us a little bit about what MOTS-c is, and let's maybe start with the discovery. I believe you were part of the group that actually discovered MOTS-c. How did you guys stumble into this? Were you looking for it explicitly, or was it serendipitous?
Yeah, so I discovered MOTS-c when I was a postdoc in Hasi Cohen's lab at UCLA. And I was really interested in aging. Of course, that was my main interest. And instead of going on to another type of postdoc, I met Hasi, who was working with the first one that was found, the first peptide that was found in the mitochondrial DNA called humanin. And it was very interesting. And the idea for me was, well, maybe there's more than just humanin, because why would there just be one? And just to give a little bit of background, my first papers as first author, my first author paper was in my undergraduate research lab, excuse me, in Kelly Hughes' lab at UW. And my background was to look for genes in the mitochondrial, in the bacterial genome. So regulatory genes, going hunting for genes in Salmonella was my first paper. So I already had this background of bacterial genetics, looking for genes that are regulators in the bacteria Salmonella. And I forgot about that for a while because I moved into humans and aging. And then I heard about these peptides. And so it kind of clicked with me, kind of a cruel twist of fate, maybe. I left the bacterial world, went into humans, and then I got attracted back into that half bacterial, half mammalian world. And I thought it'd be interesting to look for more genes. So I guess in a way, it's interesting way that science careers can develop. So I came back and said, let's look for more genes. So it's very intentional. But then if you start looking at the mitochondrial genome, even if it's small, it's only 16.5 kilobases compared to the nuclear genome, it's just very minor in size. But if you apply the possibility of genes that can be encoded inside other genes, right? Or genes that can overlap and share codes, that's very typical of bacterial genomes, then the possibility was very, very large.
Yeah.
So I started to look for papers to kind of help me guide in areas to look for it. Humanin was first discovered, it's encoded in one of the two ribosomal RNAs in the mitochondrial DNA. Mitochondrial DNA has 2 ribosomal RNAs, 13 proteins, and 22 tRNAs that were known to have been encoded. So we were looking at the map and said, well, where do we go? And it's such a large thing if you consider this overlapping gene situation. And then there was a paper that was published in Cell in 2011, what's called mitochondrial transcriptomics. So going back to the textbook drawing of the mitochondrial DNA with a very simple, layout of the genes, the transcriptomics paper said there's tons of transcripts that are small in the mitochondria that we cannot map to genes again because it was not annotated. So there were hotspots here and there. So we were looking for different areas. And then I came upon this paper that was published by Sidney Pesca's lab, who's considered the father of interferon. He recently passed away, where they kind of published this paper out of frustration in 1983 in BBRC, basically saying, hey, now we know how to induce interferon. We know that it's an immune protein. You infect a macrophage or monocyte-like cell with a virus, endovirus, and that induces interferon. They got to that part very well. And then they said, well, let's look at all the genes that are induced after interferon goes up. And those might be interferon-induced or regulated genes. Very logical, right? And this is before RNA-seq or microarrays or any of that, qPCR. So they went through restriction mapping, right? Which is basically figuring out, piecing together the sequences. And they said roughly about 80% of what they find that is induced by transcripts that are induced by interferon or induced after interferon goes up, map to the mitochondrial DNA. And there they kind of put a finger on a rough area in the mitochondrial, a loci, a locus, which happened to be ribosomal RNA. So we had humanin that was in one of the 2 ribosomal RNAs. And then the other ribosomal RNA, there were several other potential possibilities. And that's where we went to look for these genes first.
Yeah. So you sort of, a couple things were going on here. Interferon, it's an immune molecule. As we said before, you would probably expect the mitochondria to be communicating with immune system stuff. So not too surprising, perhaps the mitochondrial genome would light up, so to speak, in response to something like interferon. And also, despite the fact that the mitochondrial genome is like a bacterial genome, it's small and it's simple, sure, relative to the nuclear genome, you guys surmise that there's probably more going on there than we know about yet or we appreciate because this is biology, things are messy, there can be lots of genes interdigitated with one another, lying on top of each other. And so sure enough, you sort of discover that there is more going on here, and that brings you to the discovery of MOTS-c.
Yeah. And it's really interesting. Like I like the way you put it, this whole immune connection. I didn't understand it in the beginning. Why is it an interferon-related peptide? That was somewhere in my brain, but I really didn't grasp the importance of that until many, many years later when we started to develop these theories. And it clicked one day in one of the lab meetings. If the origin of this is, in fact, from the bacteria that infected, and they did use these immune peptides, then it could have been the first immune system that the joint proto-eukaryotic cell had, that we have now. But the nature of the communication must have been immunologic. So that's consistent with the idea that immune systems and metabolic systems co-evolved for many, many reasons. And what we see now is that MOTS-c is very immune related. In fact, we just recently published a paper describing it as an antimicrobial peptide.
Yeah, yeah. And you might think naturally, like mitochondria used to be bacteria. They're generally going to probably want to have turned down immune responses in the host cell. And that will maybe connect to other things we talk about related to how MOTS-c is affecting inflammation and genes related to the regulation of inflammation. But these little bacteria, they wanted to live peacefully or become cohabitating inside of eukaryotic cells, what became eukaryotic cells. It makes sense they're going to respond to the immune response of the larger host cell and perhaps do a lot of stuff that will tune or turn down the immune response so that they're not just killed off like we would kill off a pathogen. So let's talk about what stimulates MOTS-c and then talk about sort of where MOTS-c goes and does stuff within the cells. In general, we talk, in the literature that I've read from you and others, MOTS-c tends to be induced from metabolic stress. So what exactly does that mean? And what are the high-level stimuli that induce the mitochondria to produce MOTS-c?
So it depends on the cell type, I guess. I guess when we talk about mitochondria, and this is kind of what we talk a lot about within the academic ring, it's easy to simplify mitochondria as one thing, but in fact, every different cell type, like just like they have different energetic demands, the role of the mitochondria are many, and they respond to different things because they adapt to the local environment. For example, the mitochondria in the brain versus mitochondria in a sperm, for example, they have different roles. So what we know so far is that in immune cells like the macrophage, which I just mentioned, Sidney Pesca's lab already kind of published in '83, we kind of recreated that. We wanted to ask what Sidney Pesca saw really the peptides we discovered. And so if you stimulate monocytes or macrophages, we do see a time-dependent increase of the peptide at the protein level. So immune responses could be one that could stimulate MOTS-c production. And then another one that we published was exercise. So exercise is a combination of multiple different types of stress, right? It's metabolic stress, but also heat stress that the mitochondria generate. Your mitochondria can heat up to, according to some papers, over 60 degrees Celsius, which is basically, a death situation. So there's a lot of heat stress. And then there's mechanical stress, muscles contracting and shearing. So all these stressors will activate. So we don't know exactly what the stimulation is that triggers MOTS-c activation, probably a composite that the mitochondria sees.
Yeah.
And so that's another one that we know. Other than that, it's still in progress. We're trying to figure out what it does. In the brain, we also showed in a paper in 2021, in a cell metabolism paper, we showed that in the brain also exercise will stimulate MOTS-c production in a place called hypothalamus.
Yeah. So exercise will stimulate MOTS-c production. Other forms of stress, including immune reactions, will as well. There's probably hundreds to thousands, potentially even many thousands of mitochondria per cell. Throughout this conversation, I'll let you sort of direct us to what cells and tissues we're actually talking about for any given experiment or phenomenon we might be discussing. But in general, let's say someone's exercising, an animal, a human, whatever, that's causing some amount of stress. The mitochondria are detecting that one way or another. They're producing MOTS-c. Where does that MOTS-c go? Is it staying inside the mitochondria? Are they excreting it and it goes everywhere within the cell? Where does it go and what does it do from there?
That's the question. We're still trying to figure that out. So in the 2021 Nature Comp paper, we showed that in humans, this is a collaboration with Troy Murray at New Zealand. He had these 10 undergraduates who were very brave to give muscle and blood while on a stationary bike. So we measured MOTS-c levels before, during, and after exercise. Of course, during exercise, we couldn't get their muscle. So that was just the blood. And we do see a very significant increase of MOTS-c in the skeletal muscle as well as in circulation. So MOTS-c is secreted out in the circulatory system, and it does act as an endocrine factor, if you will, or I see it more as a cytokine now. So it does circulate. The source has always been a question. When we first published the Denu paper, the commentary kind of named MOTS-c or referred to MOTS-c as a mitochondrial hormone because it circulates. And then some people call it a cytokine or mitokine. We were leaning more towards the mitokine than a hormone because by definition, a hormone has to have a specific cell that secretes it. And we didn't think that it was one cell type or one tissue type. Many cells seem to be expressing MOTS-c from our studies. So the source of the circulating MOTS-c during exercise, especially, we don't know exactly where that's coming from. We suspect that the muscle is involved, but we haven't really found a way to detect that. In terms of the elevation of the skeletal muscle, that's the same story. We're not sure if it's made in the muscles and kept inside the muscle tissue or skeletal muscle, or if it's taking and accumulating it from the circulation. That we're not sure. But what we know is that the levels do go up.
Yeah. So levels of MOTS-c go up with exercise. They plausibly go up with other forms of stress as well, but they are definitely exercise inducible. And this is not a highly localized signal. It's not just staying within the mitochondria that produces it. It's getting to other places. It's getting into general circulation to some extent.
Yeah. The source, and we would really love to know where that comes from.
Yeah. And now we mentioned before that there is communication between mitochondria and the nuclear genome. What do you guys know about MOTS-c and when it's produced by mitochondria, whether or not it's regulating the nuclear genome?
Yeah, so in the textbook still today, the nuclear genome is described as the keeper of all regulatory genes. Things that regulate the cell are exclusively in the nuclear genome. The 13 proteins in the mitochondria, the big proteins, were just trapped there to support the electron transport chain. That's kind of how the textbooks describe this. In 2018, we published a paper in Cell Metabolism showing that MOTS-c actually does go into the nucleus upon stress. And this was work by a postdoc in the lab, Kyung-Wha Kim, who now has her own lab. But one day, she came and said, I keep imaging MOTS-c, and I'm looking at the microscope, and there are these little speckles in the nucleus. Like, it's so weird. And it doesn't seem to be an artifact because it happens, too frequently. And so we came up with this hypothesis. Well, probably then maybe MOTS-c, something in the mitochondrial genome, could go into the nucleus. And it's not new. It's very consistent with the fact that the nuclear alphabets of the language regulate chromatin dynamics in the nucleus. So, like I said, the histone regulations, the phosphorylations, the acetylations, these are mostly metabolites. Acetyl-CoA provides the substrate for acetylation. ATP phosphorylation, methylation comes from the methionine folate cycle, which half of it almost runs in the mitochondria. So it's not new. ROS, oxidative stress, is also known to regulate gene expression. So communicating with the nucleus was already an established route of I guess, communication. And so we asked the question, well, does it go anytime? And when we fast forward, what we learned was if you stress a cell with glucose withdrawal or growth factor withdrawal or even oxidative stress, about 30 minutes to 1 to 2 hours, you see MOTS-c just moving into the nucleus very significantly. And then it stays there depending on the cell type. We used a generic cell line because we thought this was more of a fundamental biology. We used something called a hectonite-free cell, which is really a very generic cell type. In that cell type, it stays in there for about 4 hours, and then it comes back out or disappears. We don't know if it's degraded in the nucleus or if it gets shuttled back out, but the signal disappears from the nucleus. So it's one of those rapid stress adaptive signals, MOTS-c, that goes into the nucleus, and it regulates nuclear gene expression, adaptive nuclear gene expression.
So some kind of stress happens. It could be something like a cell being deprived of glucose. That causes the mitochondria to make MOTS-c. MOTS-c levels go up, and then at least some of that MOTS-c goes into the nuclear genome, into the nucleus, where it is presumably causing some genes to turn up and turn down. And it would make a lot of sense that this is going to be to stimulate some kind of adaptive response to the stress, to make the cell better able to cope with that stress, whatever it may be. So another question that comes up at this point is, which genes get turned on and off? Is there a clear pattern as to how MOTS-c is regulating gene expression?
Yeah, so that's a really interesting question, and one that one of the PhD students in the lab is really digging deep for her thesis project. Long story short, MOTS-c is a very small protein, and it doesn't have specificity. So bigger proteins have specificity because there are many bells and whistles. Smaller proteins have less, but they're much more versatile in the way they can move around. They can bind to things easier, right? So what we think is going on is MOTS-c itself does not directly regulate gene expression because it's too small. It acts as a cofactor. It basically provides a flavor when the cell decides to change its policies upon, facing some kind of stress. And the mitochondria will have a say in how this should be operated, right? The bigenomic integration. What we think is going on is, and this is really interesting because it's kind of related to histones from our observations. So histones, many people believe, are derived from antimicrobial peptides. So, bacteria don't have histones. Mitochondria don't have histones. They just evolved in the middle somewhere. And if you look at these DNA binding proteins, they also operate in the same way that antimicrobial peptides work. Because I don't know if you remember when I said antimicrobial peptides have a positive charge in that hydrophobic domain. The DNA is negative. So the positively charged residues will bind or get attracted and interact with the DNA. And then the hydrophobic domains allow for interaction with other proteins. So when we talk about chromatin dynamics and histones opening and closing, and sirtuins and NAD and acetylation and all of that, what basically is happening is an electromagnetic shift in the field. So histones are using their positive charge, are binding to the DNA. If you acetylate the histone tails, that changes the local electromagnetic field and it loosens that interaction so it can open or close. Kind of the same kind of concept. MOTS-c goes in and it regulates various types of genes depending on the stress. So this is an adaptive stress, so it's not a generic stress. It's specific to the type of stress that it's trying to respond to. And the reason we think it's so diverse is because the other transcription factors that come in response to that particular type of stress will bind to the DNA. And those transcription factors are the ones that start opening up the chromatin. And then MOTS can go in, and in those exposed areas, it can bind and regulate the electromagnetic field there.
Got it. So let me see if we can unpack this a little bit for people. Inside of the nucleus, we've got the nuclear genome, thousands and thousands of genes, lots of long stretches of DNA. And DNA can be regulated, genes can be turned on and off in different ways, so different levels. So the histones and the stuff that you're referring to, they're like these spools. People sometimes use the analogy of like a fishing reel, right? You have a bunch of line wrapped around the reel.
Right.
The stuff that's exposed, the string that's exposed, can be turned on or off. It can be bound by transcription factor proteins and directly accessed by proteins and things like that. But if something's wound around the spool, it's completely inaccessible. So you can change the transcription factors that are sort of turning on and turning off the open parts of the string that are not spooled around the reel. But then you can also change which segments of the chromosome are spooled up and tight versus accessible to these proteins. And, without getting too much into the biophysical weeds here, a lot of this has to do with biophysics, positive and negative charge, things sticking to each other. A lot of peptides are biophysically well positioned to change some of these charge interactions and sort of maybe loosen up which segments of DNA are accessible to be turned on or not.
Exactly. And we still see that today in our body. One of the antimicrobial peptides we have in the nuclear genome called LL37 also binds to DNA. And there's evidence that if our LL37 antimicrobial peptide binds to our own DNA, it can present it to our immune system in an erroneous way. And there is some evidence suggesting that is the foundation for autoimmune conditions like psoriasis, where your body starts to attack your own because it's displayed. So yeah, so it's kind of in the same context.
Yeah. And you mentioned, right, these peptides are relatively non-specific by nature because they're mini proteins. They are relatively short sequences of amino acids. They're not as elaborate and they don't have as much refined structure and bells and whistles as a larger protein. And therefore, they have less specificity. So they're not acting like a transcription factor, which might be highly specific to a very small number of genes that it specifically binds to and turns on or turns off. They have these sort of more generic interactions and they're sort of more generally shaping which parts of the chromosomes are accessible to be played with by other things.
Yeah. So if you think about these bigger proteins that are like the engine of a car, it's fine tuning the response.
Yeah.
So depending on the terrain, now how do we fine tune this engine? Is it going to be a SUV? Is it going to go off roading? Is it going? It's going to be on a track? Is it going to be in a limousine? Whatever it is, the engine is there, but fine tuning its use makes it much more adaptable.
Yeah. But at a high level, it's like MOTS-c or things like that, peptides like that. They're not going to affect one gene. They're going to affect a whole number of genes. So because ultimately what a cell is, right? A muscle cell is a muscle cell and not a brain cell because of all the pattern of genes that are on and off, which determines the pattern of proteins and other things that are doing stuff in the cell. So if you've got something like a peptide such as MOTS-c and it affects many genes across many chromosomes, it's going to reprogram the behavior of the cell potentially in a big way.
Exactly. And these high-level regulators, so to speak, like sirtuins, for example, they regulate histones, but they don't always regulate one histone in one region, one locus of a chromosome. It provides an overall shift, and that makes perfect sense because when a cell goes in to regulate nuclear genomes, and from the mitochondria's perspective, it's a really one of the most sensitive sensors of the environment. And the mitochondria is trying to say, hey, look, our funding levels are going down. The economy has to shift. And to adapt to that, you can't just go and pinpoint to one thing. It's an overall code red that every department has to respond to, right? Because the overall economy is going down. So it makes perfect sense that we kind of say, the mitochondria talks to the proto-mitochondria. The bacteria was trying to communicate and regulate the bigger cell, but the bigger cell was counting cash, the rent. So these ATP, NAD, these are actually cash currencies in the cell that pay for processes. And so they directly read off of the economic state and incorporate into their gene expression profile, which is very smart in a way because you need to have an overall sense of the economy. And so histones, instead of being specific, it's much more strategically beneficial, increasing the fitness of the cell to make it a more broad, general regulation.
Yeah. And then hopefully by now people are starting to appreciate how powerful this type of regulation could be, because immediately you can think of all of the situations where there's some kind of stress that requires a pretty wholesale transformation of what our cells are doing. Like if I get sick with an infection, right, I've got to have white blood cells that essentially transform they go from, a dormant state where they're not doing so much to a completely different state where they're attacking and directing resources and dealing with the infection. If we exercise or we fast and our cells are running out of energy or energy demands are changing or we're running out of fat stores or glycogen stores, our cells, whether they be muscle cells, fat cells, or whatever, they might have to completely change what they're doing in many ways. So they're going to need to turn on and turn off whole constellations of genes. And that will require some kind of signal, such as MOTS-c or one of the things that we're talking about, that can have that sort of general reprogramming effect rather than just turning on one gene or moving one protein around.
Exactly. And also the component of time as a biological variable, I think is very important here. So because we're more focused on aging, the way I see, and many people in the field kind of look at bioresearch, is that there's a unbalanced focus on acute responses for many reasons. Nobody wants to do a long experiment, and you want to understand the mechanism. So a strong induction is required to understand pathways in the beginning, right? When we don't know anything. But in fact, what really happens in our daily lives is more of these subtle, moderate stress that's more chronic, that's more relevant to us. Unless you have a disease situation or some kind of infection or accident, most of the stress adaptations are going to occur over time. So, for example, exercise is what induces MOTS-c, but for the same reason, MOTS-c is not going to be a acute, regulator. It requires time. So you need to go to the gym for 6 months plus to see some change in your body, because your body is adapting to that. And that's very smart, because nobody wants to shift their policy dramatically, with a cough or with a temporary transient stimulation.
Yeah.
That's just not good.
Yeah. And before we discuss some of the phenomenology, the amazing phenomenology of MOTS-c and some of the early papers you did, I want people to also think about, our cells need to transform what they're doing and how they're behaving all of the time. And that can happen in healthy or adaptive ways. Like you can turn on immune cells so they deal with the problem and then they turn off. You can shift the bioenergetics of your cell to do your exercise, and then you rest and recover afterwards. And things are constantly changing and waxing and waning. But then, of course, there are disease states, pathological states, age-related decline states where the cell might get stuck in a state it's not supposed to be in, or that is suboptimal. And we want to figure out how to unstick it. You mentioned things like autoimmune conditions like psoriasis. I actually have psoriasis. In a sense, you could think of it as, the skin cells and or immune cells are somehow stuck in a state of hyperactivity that we would like to unstick them from. And then when we start to think about these peptides like MOTS-c as these sort of general purpose metabolic reprogramming signals, they start to become very interesting and potentially very powerful from a therapeutic standpoint when we start to think about shifting our cells and tissues out of pathological states and into healthy states.
Yeah, I love the way you phrase that. So maybe I'll start with this in response to that comment. Drug development historically has been exactly that. It doesn't cure the disease, but what they're trying to do with these drugs is to be very specific in correcting what has been wrong, with the goal of bringing back homeostasis, right? The balance. And of course, this country's medical system is the same way. FDA approves drugs for disease indications only, right? With a very specific mechanism. But the problem, again, like you were saying, is bringing back the balance is a very generic thing, a general thing, if you don't have a specific problem, right? So what do you do in this situation where if you want to extend the person's healthy lifespan, you don't wait until you get a disease and try to treat that and then therefore artificially prolonging their life. What we want to do is to really increase our homeostatic balance. And that's exactly what we do with exercise, right? Every time you go and exercise, you're slowly increasing your capacity to maintain homeostasis in a very stressful condition, right? So that takes time. And for that reason, a lot of these regulators will be general and multi-component, multifactorial shifts. It's a big problem because it's not going to be therapeutic in a sense that it's going to target the traditional way of approaching the disease, but therapeutic in a way by increasing our youthfulness, which I define it as the capacity to maintain homeostasis, just like we do with exercise and diet. Yeah.
Let's talk a little bit now about some of the effects of MOTS-c that you've seen in animals. And, people should keep in mind some of the mechanics we were kind of discussing before. MOTS-c is this peptide. It's a microprotein. It's made by mitochondria. It's stress responsive. When we exercise, when something happens like that, the mitochondria make MOTS-c. It can go into the nucleus. It's changing the expression of a lot of genes to change what the cells are doing. So let's now talk about some of the interesting experiments you did in mice related to obesity and insulin resistance. If we go back to that first MOTS-c paper that you did, where you're looking at how MOTS-c affects metabolic homeostasis, broadly speaking, what were the basic results you saw in that study with respect to diet-induced obesity and insulin resistance?
So when we first found MOTS-c, we had no idea what it did. There was a very loose expectation that it had to do something with metabolism because it's from the mitochondria. And so the first cell culture in vitro experiments were to look at the effects of MOTS-c on respiration and all these metabolic parameters. And there were significant changes there. So that was convincing that it was a metabolic regulator of sorts. And then when we moved into mice, we still had no clue, because how do you translate a cell culture experiment into a physiological condition? So we went with the thing that we knew best to regulate metabolism in mice, in Hassi's lab. Hassi is an endocrinologist by training. And so diabetes, obesity, that was the first venue. So we just fed mice a high-fat diet. We knew that was a metabolic stress, and that screws around with their insulin sensitivity and all that. And then we started to inject MOTS-c to see if it does anything, either make it worse or make it better.
So the basic setup here is you were doing diet-induced obesity models in animals, pretty similar to many, many other experiments that have been done. You give them a certain type of high-fat diet. We'll not get into the details of the nutrients there in this podcast probably, but you give them a diet that makes them fat. They become bigger, they become fatter, they become insulin resistant. And the experiment is, okay, if we do that, but we give them MOTS-c, what happens?
Exactly. That's what we did. And we just basically observed what happened. And it was really shocking because it reduced, or it prevented diet-induced obesity very dramatically. And when we were dissecting the mice, you open it up, and one of the characteristics of these high-fat diet mice, and the same for humans, is fatty liver. You open up the mouse, and the liver is clearly pinkish because of the fat accumulation, where it should be very deep red. And when we opened up the MOTS-c ones, not only did they not gain as much weight, their livers were all red. And so that was the beginning of the studies that led to MOTS-c into translation into clinics. So a company called Cobar, which was co-founded by Hassi Cohen, my postdoc advisor and the dean of our school right now, and also Nir Barzilai, who's in New York. The indication that the company went for was fatty liver and obesity for that reason. So that was one of the first shocking things that we saw. And then everything else, came history.
Yeah. So you give mice this high-fat diet, they get bigger. They get quite a bit bigger, too. It's not a subtle effect. They get quite a bit fatter than the mice eating the normal diet. They get a fatty liver. If you give them that high-fat diet, but you're also giving them MOTS-c at the same time or a little bit ahead of time, they basically don't, it's not that they don't get as fat. You basically almost completely blocked the diet-induced obesity. It was a dramatic effect, yes?
Yes, it was it wasn't 100%, but it was very, very significant.
Yeah. And then you open them up and you're also blocking the fatty liver. What happened when you gave normal mice MOTS-c? Did they waste away and lose weight?
No, in fact, nothing happened. And that's one of the themes with MOTS-c. It doesn't have a job unless there's a job to do, which means in a peaceful condition, well, as best as we can define, even in cell culture, if you don't perturb the cells and leave them in their happy state, MOTS-c has really we never saw anything happen to happy cells. It's always some kind of stress that was required.
So it's a context dependent effect. It could have been the case that MOTS-c was just causing, the wasting away of fat tissue and lean mass or whatever, but that's not the case. It was actually preventing diet-induced obesity in the context where you're stressing the animal with this high-fat diet, but it wasn't doing much of anything under normal baseline conditions.
Nothing.
Interesting. And then, when you dug under the hood a little bit, what is going on that underlies this dramatic effect? I want to ask you about sort of the metabolic cell level effects of MOTS-c. And this is going to bring us to something called AMP kinase. What is AMP kinase and how does MOTS-c tie into that?
So AMP kinase is also dubbed as the master metabolic regulator. The AMP part of AMPK is the monophosphate version of ATP. So, the phosphate in ATP is the source of energy. When you cleave that, you get energy, and you can do things, you pay for things. Once it uses the stored energy, phosphates, it becomes ADP, diphosphate, or monophosphate, AMP. What AMP kinase does, it's a protein that senses the levels of AMP versus ADP. So how much of our cash currency have we used?
Yeah,
If AMP levels go up, that means we've used up a lot of our resources. And that's more consistent with a fasting state or energy depletion state.
got it.
So the cells
So
sense.
it's like a
Go,
fuel
go.
gauge or like almost like a detector of when your bank account is running low on cash.
Exactly, exactly. It's like an alert system. It's like I guess not an alert system. It's a sensor of cash flow. And when it goes up, when AMP goes up, your cash is low. So they have to rearrange things and say, okay, look, we have to kind of live with this new environment where we're not as affluent as before, strap down things.
Yeah.
Uh, and the kinase, K part is kinase. Kinase basically means a protein that goes and phosphorylates other proteins. So it's a regulator in that
Yeah.
sense.
So AMP kinase becomes active when ATP levels go down, which basically means you're running low on cash. So if we just play with this analogy a little bit, if you're running low on cash and your income's constrained, you're not going to be building a new kitchen in your house. You're not going to be buying a new car. You're going to want to preserve your cash flow and focus on new ways to generate income.
Absolutely. Yes, yes. And so that ties in really importantly with aging for us because, one of the most important things that can regulate somebody's lifespan or health span is metabolism. And so there's an ocean of literature on how things like AMP kinase, there's another master regulator called mTORC1. That's another one that regulates more protein side. These have been studied extensively in the field of gerontology, aging. And if you activate those pathways, it typically leads to extended health span or lifespan.
Yeah.
So that
Yeah.
regulates metabolism.
Yeah. What are some of sort of the natural stimuli of AMP kinase? What makes AMP kinase go up at the level of behavior and macroscopic stuff?
Very clearly, energy deficiency. So dietary restriction will definitely increase it. But this is one of the things that I think most people that are not in our field kind of go over and miss is that these are stress signals and they're not supposed to be activated for a prolonged period of time. So think of it as stress hormones like cortisol. Yeah, you need cortisol, but you don't want cortisol levels up for extended period of time. It should spike and then go back down, right? Because it activates downstream pathways and then it has to shut off. Otherwise, if it's constantly up, that causes your demise instead of
Yeah.
saving
Yeah.
you. So
Yeah.
it's hard to say what activates and what goes, but
Yeah.
clearly P,
Yeah.
energy
Yeah.
deficit.
But the things that would cause that: fasting, food deprivation, exercise, and as you're sort of implying here, these things can be good or they can stimulate this pathway in adaptive ways. But you don't want these signals to be turned on chronically or for too long, right? You don't want to exercise too hard for too long because then it can go from being a healthy thing to an unhealthy thing. Fasting can be great, but obviously if you go too long without food, then you're in trouble.
Malnutrition, yes.
So AMP kinase basically gets activated more when MOTS-c goes up, which can be induced by exercise and other things. What happens after that? What happens to the mitochondria after that? What's sort of the net result of turning up this AMP kinase pathway in terms of the mitochondria and what they're doing and how many there are?
So many, many things happen, including AMP kinase activating a pathway through something called PGC1 alpha to increase mitochondrial biogenesis. So it's an energy deficit situation. So you want to maximize your energy efficiency. So it could stimulate mitochondrial biogenesis. It regulates a lot of the proteins that are related to metabolism, like fatty acid metabolism, it starts to increase fatty acid metabolism because it's your storage, right? And in times of deficit, you want to start using your storage. So fatty acid oxidation can go up. Your usage of glucose goes down. So a good manifestation or good example of this in a more translational way is a drug called metformin. It's the number one frontline drug for diabetes, and it regulates your glucose production and in some ways consumption too as well. So it's a broad effect because it's such a high level regulator. So if you switch around your glucose metabolism, your fatty acid metabolism, the mitochondria is at the center of that, and it'll respond in many, many different ways.
Yeah. So we're in a state of energy deficit because we're exercising or food deprived or something. MOTS-c is being produced by the mitochondria. This is causing AMP kinase to go up. This is sort of our sensor of energy deficit. We're in a state of relative energy deficit. AMP kinase is going up. One of the things this ultimately leads to is mitochondrial biogenesis. So the cell's basically saying, we're running low on ATP. Let's build up our ATP production capacity so that we can counteract this deficit we're facing. What's happening to other things the cell might be doing? Things like mTOR, the anabolic parts of what the cell is doing to build up other structures.
Right. So AMP kinase and mTORC1 are related. They're networked, so they cross affect each other. mTORC1 is a different thing in terms of benefits. Well, I guess you want to turn on AMP kinase, but you want to turn off mTORC1 because mTORC1, like you said, is anabolic. It stimulates protein synthesis. And that's not something you want to do when energy is low because protein synthesis is one of the most expensive processes in a cell. It can take up to 30% of a cell's energy source. Yeah. So when AMP kinase goes up, typically it suppresses mTORC1 activation.
Yeah. So again, if we play with our analogy, it's like if you're running a household and you get demoted at work or you lose your job, income is going down. So you want to preserve, the cash flow that you have. Think about ways of building up your income capacity, and you want to turn off the remodeling of the kitchen. You don't want to hold off on buying a new car and building up these new structures because income is constrained and those things cost money.
Exactly.
Yeah.
So you're basically strapping down.
Yeah.
Getting ready for war.
Yeah. So okay. So AMP kinase stuff we just talked about. We mentioned earlier that MOTS-c goes into the nucleus and it affects gene expression in a big way, many different genes, not just one or two genes. You're reconfiguring what the cell is doing and how it's using its energy and all of these things. Where does the methionine folate cycle come into this? How is MOTS-c affecting that cycle? And how does that cycle relate to the gene expression stuff?
Yeah, so that's what we're really trying to figure out right now. What it does in the nucleus is just so diverse. Like I said before, it doesn't have specificities. It really depends on the other cofactors that come in or the other transcription factors that come in. Yeah, I mean, that's an ongoing project in the lab. We're still trying to figure that out.
Got it. But one of the things I wanted to get you talking about is you mentioned earlier the importance of some of these phenomena, some of these signals being acute and not chronic. Exercise is a stress, and it's good to intermittently have that acute stress, but you don't want to have chronic stress, too much stress for too long. That can lead to problems. When we start thinking about exogenous MOTS-c, and we start thinking about, the natural dynamics of endogenous MOTS-c, it goes up, it comes down because stress is intermittent, it's not chronic usually. What might start to happen or what problems might start to arise if you were just, say, giving an animal exogenous MOTS-c for a very long time, turning on this AMP kinase pathway? Would it have benefits initially that maybe turn into problems with chronic use?
I mean, I think theoretically it's possible. What we did in the 2021 paper, in the Nature Comp paper, was we injected mice, old mice and middle-aged mice, old equivalent to humans of roughly 65 years of age, 60, 65. We injected the mice with MOTS-c daily. And within a week, we saw a trend of physical capacity going on, meaning they were running much longer on the treadmill. And then, but it wasn't significant statistically. So we extended the protocol to 2 weeks of daily injection. And then the older mice were doubling their running time treadmill, doubling. And they were outrunning their middle-aged friends, or I guess they're not friends, they're clones. So they were performing really well.
Yeah.
And the middle-aged were doing well too. But these were daily injections. So what we did after that was the effect was much stronger than we anticipated. So we didn't want to euthanize the older mice. They're very precious resources. So we switched the protocol and got approved to do lifelong MOTS-c injection. And here is what you were just saying really comes into play about this stress signal that shouldn't be up the whole time. And that's true for a lot of these longevity interventions. Like, for example, rapamycin, which is one of the most powerful lifespan enhancing drugs out there being tested. There's a very famous paper that shows intermittent rapamycin injection is very powerful, meaning you don't want to yell stress the whole time. You want to provide that stress, give the cells time to recuperate, increase their capacity, just like exercise, little by little, and respond. So that's the major theme of MOTS-c right now. In a paper we recently put up on bioRxiv, we try to make that point very clear. And there now we have a much more molecular description of that. The MOTS-c's effect is not unimodality, meaning it's not just one thing that it does, like AMP kinase activation. It seems to be activating a program. Mm-hmm. And program has an acute phase, which is in the cell type that we use, again, the generic hectonite freeze or the fibroblast, it lasts about less than 24 hours. And that focuses on regulating protein translation. So if you take a step back and look at it, what we see, how we interpret it is when MOTS-c goes into the cells, it's a stress condition, but you don't know exactly what's going on. Like the cell's been hit with something and they're trying to figure out what it is so they can mount the appropriate response. And they have to figure out if this is going to be transient or persistent. And so the first 24 hours is kind of like the cell trying to figure out what's going on. And as we just talked about, the first thing it does is to reduce protein translation. So MOTS-c goes in and translation gets shut down very, very fast, very, very quickly. And that lasts about 24 before we see gene expression happen at around 24 hours. So it's a phase.
Yeah.
We call it an acute phase and the chronic phase. In the chronic phase, you have gene expression coming out, but this effect goes away if you don't continuously give MOTS-c, meaning it's not one-time activation. It fades away if you don't give it to it. And in this paper, we show something really crazy, and this was another crazy moment that gave me goosebumps. We use something called a PSM model. It's progressive stress media. And going back to what we were discussing earlier, a lot of the cell culture models are geared towards acute stress that are typically not physiological, meaning we take away glucose, we take away certain amino acids. And that's very stressful, but that rarely happens in a living body, right? You don't have no glucose, you're dead at that point, or an amputation or something. So what we did was we had the cells use up the media the way they do naturally, physiologically. And then we collected the spent media. But we had different gradients, spent a little bit, spent moderately, and extensively.
Different levels of stress.
Different levels of stress. And we identified it using mass spec, metabolomics. We kind of know what changed and what went up and down. And in that media, it's really comprehensive because it's what really happens in a body. It's not just usage and depletion of nutrients. The cells, while they're responding to the nutrient deficit, start making other things because it's metabolism, right? You metabolize things. So it's a very, in our opinion, much more physiological stress. And the reason that's important is because our cells have evolved to generate these programs that adapt to that situation more so than removal of a single nutrient situation. So the programs that get activated in the chronic physiological state will not be activated by, removal of certain nutrients was our thinking. And we kind of put that in the paper too. When we just dilute the media, not, not collected from a spent situation, MOTS-c does not protect the cells. It doesn't have much role, because I guess those are not physiological situations that are wired into the cell to activate this response. But when we use the spent media, normally cells will start dying off in this moderate stress, physiological stress level in about 3 days. The cells can't do more. They just go down and then die about 3 days. MOTS-c, if we treat them with MOTS-c, we kept them alive for over a month, which is crazy if you think about it. We didn't give them any food.
Right.
We didn't replenish any media. We just gave MOTS-c, and they go for over a month. And we stopped in a month because the PhD student had to deliver a baby. So she stopped there. But I don't know how far it could have gone. But
Wow.
That's where it is. But if you stop the treatment of MOTS-c, the curve starts to drop almost immediately.
Yeah.
The next couple of days after.
MOTS-c is somehow reconfiguring the cells so that they can survive in those deprived conditions for much longer.
Right. And that's why I think it's much more consistent with the exercise secretion. So exercise stimulates MOTS-c because it's preparing for a more persistent stress condition to respond to. So if I do one bout of exercise, I'm not going to get physiological changes I wish I did. But going to the gym once, nothing really happens. Going to the gym for 3 months, nothing seems to happen. Because for the body to respond to that immediately is a waste of energy, right?
Right.
Grow muscle, then it better be something that you're going to use, or it's a new environment that you have to adapt to that's persistent, right?
Yeah.
So that makes sense.
Yeah.
That MOTS expressed during those conditions.
Yeah. And a lot of this stuff at sort of the molecular and cellular level can hopefully start to get people to make more sense out of what happens at the whole body level when we do things like exercise. So you have to do things that are persistent, but also at the same time dynamic. Like if you start going to the gym 3 times a week for 3 months, and you exercise hard and then you rest, and then you exercise hard, then you rest, that's going to be different than not doing that at all. It's also going to be different than going and exercising hard every single day, which might put you into this chronic stress mode that actually prevents you from building muscle, translating protein, turning on mTOR, all of the stuff that we're talking about under the hood here. Exactly.
So it's not pressing a button to get to the next level. Our body is, again, the whole focus is on homeostasis. And the only reason you would increase that capacity is when it's necessary, because your body is very economic. It's not going to build muscle if it doesn't need it. Right? So if you put a cast on, I don't know if you had a cast before, for about 3 months, you take it off, it's almost like your arm wilted.
Yeah.
Atrophy happens and muscle is lost because you're not using it. The body doesn't need it. It gets rid of it. But if you want to build muscle, you better need it. So it has to be something that's persistent.
Yeah. If we go back to the diet-induced obesity animal model, you said that MOTS-c, exogenous MOTS-c, giving it to animals that are going to get a high-fat diet, it basically prevented most of the weight gain and fatty liver that diet would otherwise induce. What other changes did you see in those animals with regard to things like energy expenditure and just their basal metabolic rate?
So we put them in metabolic cages, and nothing really popped out that could easily explain the prevention of weight gain. For example, we were hoping that there would be, more activity, but their daily activity levels were about the same. What did change was their metabolism during night. So these are nocturnal animals. That's when they feed. So when we're asleep, they're active. And during the night phase, when they're actively running around, then their metabolism kind of differed from the controls. But, that was pretty much the only trend that we saw. We didn't see much in body heat, but then the equipment was not really there to accurately measure body heat. There was a paper that came out that showed that MOTS-c increases the capacity to maintain body heat. It's a very gruesome experiment. We were thinking of doing it, but we didn't have the heart to do it. You put mice in a cold room, in a fridge, and the body temperature goes down fast, right? if that group that published, they gave MOTS-c and they showed that the body temperature drop was much more retarded. It maintained body heat much more. So one hypothesis that we had was there are many ways or many reasons that MOTS-c in that obesity study that we did prevented weight gain. One of them could be expelling the energy through heat,
Yeah.
but it's not going to heat up so we can detect it easily. It's going to be a subtle increase over time, but it's going to be difficult to detect.
Got it. Got it.
So that's our suspicion at this point because of
Yeah.
that study.
Yeah. Yeah. MOTS-c has been called, it's being called an exercise mimetic. Like, you take exogenous MOTS-c and it's going to be as if you exercised, but you didn't actually exercise. Is that a fair description? Is it recapitulating most of what happens with actual exercise or just a piece of it? What do you think people should?
No. But I think, I mean, I hear that a lot too. I'm not a big fan of the word exercise mimetic, just because I feel that discounts the complexity of exercise and kind of frames the way people think of exercise as a single dimension
Yeah.
activity. If you think about it, exercise is not just about burning calories. It's also about innervation of the nervous system. It's also about connecting the nervous system with the muscle, because one of the reasons you don't grow muscle is not because of protein intakes of the deficiency. It's also your nervous system has to know that it's being used to activate it. Like I said, if I don't use my arm when I have a cast, I'm not using it. My brain is not connecting to it, so it gets rid of it. So the connection to the nervous system is one thing that's totally missed. And then the complexity of the stress. Like I said, it's heat stress, it's shear stress, it's the entire universe stress, right? Everything that happens, but not just biochemical. So to recapitulate all of that with one factor, I think is going to be very difficult. Is MOTS-c a component? I definitely believe it's a component, and perhaps it falls into the category of exerkines. These are those immune responses in response to exercise that triggers these physiological results. In 2021, we published a paper in Cell Metabolism showing that. So in the brain, the hypothalamus, which is the part that regulates metabolism, there are cells called POMC neurons that express this protein called POMC that regulates appetite and energy expenditure. What we see was if you put the mouse on this exercise regimen, which is basically a treadmill, moderate exercise, this is not crazy athletic, this is just very moderate exercise, you do see these exerkines like IL-6 go up and they end up in the brain. So the muscle is talking to the brain when you exercise for a while. And these are immune responses as well, interesting enough. And the brain receives that and it starts regulating things. MOTS-c levels go up in the POMC neurons and it works with these other exerkines like IL-6. So IL-6 will signal through its receptor and MOTS-c will interact with something called the STAT pathway. And it promotes the expression of these POMC genes, which then gets processed as regulators. The other interesting thing that we saw was when we inject MOTS-c intracranially, so if you go straight into the brain and inject MOTS-c into that part of the brain, what we see is a dramatic shift in the peripheral metabolism, especially with the white adipose tissue beiging significantly. So you see an increase in beige fat when you inject MOTS-c into the brain, not peripherally.
So you inject it into the brain and you're seeing effects peripherally. So somehow the brain is changing and sending signals out to the body. What's the effect of the beiging of the white adipose tissue? Or excuse me, what's the significance of that effect?
So, I mean, beige fat has many things that change the physiology. So it's changing, for example, white adipose tissue would be your fat storage into a much more brown fat-like condition. So brown fat is the fat that keeps you warm.
It can generate heat.
Yeah. We don't have much as adults, but babies have much more. But we still have it in certain areas like the heart, where you have to have that energy source and heat. It increases basal, usage of these lipids because of their characteristics, phage fat.
Mhm. Got it.
So
So it's
heat
like,
generation
yeah,
from there.
MOTS-c can get into the or it's going into the brain, or when it's in the brain, it's having effects throughout the body and basically stimulating changes in other tissues, even though it's the origin of the signals in the brain.
Right. And then the POMC neurons, they're related to endorphins. So
Right.
the POMC gets processed into endorphins. So we were kind of joking with the PhD student who did the exercise study, and we said, you know what? I don't think we're really doing that much to the mice other than making them high. So they're runner's high. I don't know if you've heard that term. Runner's high is basically endorphins kind of easing the pain in these elite runners so they don't feel much. And we're saying, maybe the mice were running longer because it's a pain management system. We're activating this metabolic pain management system. So we were looking into that a little bit, but we haven't gone far in that realm.
Oh, interesting. I mean, quick side note, I believe there's work out there showing that this effect in mice is actually mediated by endogenous cannabinoids, and not, yeah.
That's the same pathway, yeah.
Yeah, okay. Interesting. I want to ask you a little bit about autoimmunity. We talked about immune system modulation by MOTS-c and other peptides. There's one paper that looks quite remarkable that you were involved in using MOTS-c in an autoimmune diabetes model. Can you talk a little bit about what you guys saw there and how significant that was?
Yeah. So type 1 diabetes model. So type 1 diabetes is an autoimmune disease, unlike type 2 diabetes. And those two shouldn't be mingled too much in terms of, models. It basically is your own immune system attacking your beta cells, your pancreatic beta cells that produce insulin, and then you run out of insulin. It's a very painful disease, right? The only treatment for that is to keep injecting insulin. What we saw was in the model, we prevented the onset of type 1 diabetes, the autoimmunity. And when we looked at the pancreas, specifically the beta cells, the infiltration of the immune cells that were typical, that come in to kill your beta cells, were not there. So the immune cells did not come. They were not recruited in the beta cells. And that's why we think the pancreas was kept alive and it was making insulin as usual. What we kind of figured out was the involvement of the immune cells and the regulation of immune cells, more specifically in that paper, T cells. So an experiment that the first author did was, if you take MOTS-c treated beta cells and immune cells, and if you give it to another animal, meaning you take a normal mice, and then you take the immune cells of a type 1 diabetes model. So the immune cells are now transferred into a normal mouse. So you'll have antibodies or immune cells that recognize yourself as a target. If you do that in a normal cell, you'll induce type 1 diabetes there too. But if you take the immune cells from the MOTS-c treated type 1 diabetes mice, right, and you transplant that, you transfer that to a normal mouse, nothing happens, which tells us that it suppressed the autoimmune recognition somehow.
Yeah.
And it turns out that it has to do with something called regulatory T cells. So the T cells that actually regulate the autoimmunity. So if your antibody or if your T cells recognize your own self, these Treg cells will typically come and say, no, that's you, don't attack.
Yeah.
Those cells were much more involved, or MOTS-c was those cells.
So you've got an animal model, a mouse model. They will develop autoimmune diabetes, type 1 diabetes. Their immune cells will attack their pancreas and destroy their insulin-producing cells. If you give those mice MOTS-c, exogenous MOTS-c, it prevents the development of that autoimmune diabetes. And what seems to be happening is, one way or another, MOTS-c is affecting how some of the white blood cells, some of the T cells are developing and changing so that they don't develop this pathological tendency to attack the animal's own pancreas.
Exactly. And exactly how that happens, we're not sure, but we believe it's going to be not a direct action. Of course, there are direct actions on the actual T cells that go through the mTOR1 pathway, as we figured out. But I think, broadly speaking, it's not just the T cells that are affected. We have evidence now that the macrophages are also infected. So these are antigen-presenting cells, macrophages, B cells. And what we think is going on is that it's shutting down the activation of these immune cells. meaning it's regulating how the cells decide whether or not to, stimulate a further path or a further response. And that's why it's reducing overall autoimmunity.
Wow. Does this potentially imply that one could change whether or not autoimmune diabetes develops based on the pattern of stress in early life? So, for example, could an individual exercise more in early life and potentially affect the trajectory of such an autoimmune disease?
So that's the idea. So with aging, autoimmunity goes up, especially in women. And so if you push back the process of aging or slow it down, yes, the idea is that you will also push back the onset of these age-related autoimmunity. And one of the projects, so that's kind of what we call immune tolerance project. That in our lab, there's a PhD student who's digging into that as well as for a thesis. What MOTS-c seems to be doing is to, again, provide the flavor to these macrophages when they're deciding whether or not to activate things. But what we found in the paper, we showed in the paper, was the way that it does it is by shifting the way the macrophages mature. So if you rewind a little bit, macrophages, there are two types. They're the tissue resident ones that come from a different lineage. And there's the monocyte-derived macrophages that come from your hematopoietic stem cells, like other immune cells, right? When the monocytes differentiate into macrophages, it's taking cues from all over. For example, is this an infection or is this an exercise-induced repair that we need to do? Muscle shearing, that's what happens after you exercise. The immune response is very dramatic and it's required for muscle building. But that's more of a repair kind of macrophage that needs to clear out the debris of the broken down muscles.
Yeah.
Signal repair. Versus an infection, your job is to go and kill. It's a license to kill kind of situation.
Right.
So the gradient of different types of macrophages is very, very broad. There are different types. What MOTS-c does when we as we showed in the recent paper in eLife, when we give MOTS-c during that monocyte differentiation, it provides a different kind of macrophage, which is really interesting because if you go back to the story of MOTS-c discovery, we were basing a lot of that on that Sidney Pesca paper with the interferon.
Right.
And the most striking thing is the signature of the macrophages that are made with MOTS-c as a cue is defined by two things. One is interferon, and the other is antigen presentation.
Wow. Yeah. So you're shifting, MOTS-c is shifting the pattern of white blood cell identities that show up.
Yeah. So broadly put, and this is just my interpretation, instead of saying, hey, there's an infection, you have to go and kill it. It happens to be your pancreas. The mission changed because they're saying, no, it's more like a muscle repair system. Let's not go and kill it. But let's figure it out. What's going on.
Yeah, a cleanup crew as opposed to the Navy SEALs.
Right, because you can recognize exactly. The cells can still recognize yourself as a foreign thing. I don't think MOTS is changing that. That's already determined during development. But what it can do is to change the mission.
Yeah, change the mission and therefore who's called in to achieve it.
Right. You recognize it, but bring your tolerance out. This is not an infection situation. Let's call it a repair, environment.
Yeah. So one of the things that people who are injecting themselves with exogenous MOTS-c are reporting, the most common thing that I see out there is basically body composition / exercise related changes. So people are saying that it's helping them reduce or burn off body fat, but maintain muscle mass. So if they're working out, lifting weights, they're not going to lose their muscle mass, but they are going to lower their body fat a little bit. It seems like a lot of people are, assuming they're getting genuine MOTS-c, and I'm not sure how people are dosing this, but it also seems common that they're cycling this, so they're not taking it for weeks on end. As far as I can tell, they're often taking it for a couple days on, a few days off. They're doing things like this. Given all of what we know from the preclinical research and the mechanistic work that people like you have done, is this a likely outcome for humans who might use exogenous MOTS-c? Saying nothing about whether they should or shouldn't or what the rules and regulations currently state. Does that make sense to you that it would be preserving lean muscle mass but helping them burn off body fat?
Yeah, yeah. It's very consistent with the preclinical data, and uh, it's difficult to ignore the fact that exercise is, to the best of our knowledge, one of the strongest inducers of MOTS in humans. And therefore, a lot of the effects of exercise would be seen with MOTS is my expectation. And it's not a mimetic, it's part of the exercise response. And therefore, you expect. But like I said, the effect of exercise is many. Of those, I believe MOTS-c would have an impact on the immunometabolic side of exercise. So the benefits that we see with immune system changes, as well as the metabolic, especially the conjunction of the two, the composite effect of the immunometabolic response is what I would expect to see.
Yeah.
Are you burning more fat is a good question. Our data indicates that MOTS-c doesn't really increase beta oxidation. But the way that it's reducing fat accumulation, so there's two different things. There's the burning of the fat, and then there's the reduction of synthesis of fat storage, right?
Yeah.
So what we think is happening with MOTS-c is more on the lipid regulation, and we put this in the bioarchive paper a little bit. Under stress, we learned that one of the most important, this is already known, we just learned recently, after learning about it in textbooks for a long time ago. One of the most important things with the cells during stress is to retain its membrane lipids.
Yeah.
So other things can be sourced from various nutrients, like energy can be from multiple different sources. But the one thing that the cells cannot give up or go wimpy on is the lipid membranes. So that includes internal organelle membranes. Like if you want to increase autophagy, you need to have the vesicles, the autophagosomes, which are all lipid, right? And then the cell membrane is lipid. You can't give that up. What we see is not MOTS-c burning fat during the spent media condition. They're not using the fat to generate energy. That is not there in our metabolomics. But what is very clear is that they're shifting the lipids into phospholipids, which is not the type of light fat triglyceride storage form.
Yeah. Membrane lipids as opposed to adipose tissue.
Exactly. So what we think is that it's something like the ketogenic diet. You're not burning fat. But the fact that you don't have carb, you don't activate the mechanism of the lipases to start putting in more triglycerides into your body, right? That's why you lose fat on a high fat ketogenic diet, right?
Yeah. So it's not the burning of fat for energy. It's where the fat's going and how it's being utilized.
That is, to our knowledge, what seems to be more so.
Yeah.
I mean, I'm not saying it doesn't burn any fat.
But in your model systems, that's more so what seems to be going on. So you also mentioned earlier, we talked about AMP kinase and mitochondrial biogenesis. You're basically boosting how many mitochondria you have. In older animals, in your guys' hands, it could double their exercise capacity on the mouse treadmill stuff that you used. I suppose one effect in humans could be that to the extent it is increasing exercise capacity, maybe people are just working out a little bit harder when they're taking MOTS-c or something like that, rather than burning off fat.
So yeah, I agree with that, and I met so many people that are taking MOTS-c recently. I actually sat down next to somebody on a plane who was taking MOTS-c, and that person was looking at my presentation. I was doing my presentation in the plane, and they asked, "Oh, do you know MOTS-c?" And I said, "Well, yeah." And they told me their experience, which I think is consistent with what I've heard a lot from other people. But basically, it doesn't make you superhuman. It doesn't make you stronger or faster. What people kind of feel is that the recovery time between the intervals or the exercise goes down, which means it's easier to get back on, which is kind of what is consistent with the immunometabolic side of these things. The macrophages that go to your muscle, they have to signal and clear out the debris.
Yes.
And they have to resolve that so you can go back and do it again.
Yeah,
right?
yeah.
I think that's what's going on with MOTS-c.
Got it. So it could be that, yes, it's allowing for sort of more efficient recovery. If all of these things are true and sort of the results you had in the diabetes paper what's happening in humans who are exercising. It could be that you're shifting the profile of immune cells that come in the recovery phase, so that you're just cleaning up the debris more efficiently and not calling in some of those other types of immune cells that might not be aiding in the recovery.
Right. In a way, clarifying the mission.
Yeah.
Uh, and,
Yeah.
and that's why in the older mice, when we injected them and they were running better, we were trying to figure out why. And it came down to the muscle. That seemed to be the tissue that really responded, but also the liver as well, because there has to be a metabolic cross communication. But in the muscle, what we found with all these unbiased omics is that it was regulating proteostasis. So we were really hoping to see a huge shift in metabolism or mitochondrial, whatnot. But the signature was very clear. It was maintaining protein homeostasis as one of the main mechanisms.
Yeah.
So it's multiple things. The immune cells are involved. Like I said, MOTS-c being a mitochondrial thing will have multiple roles in different types of tissue. So from an immune perspective, yes, the recovery with the clean out and the signals to repair. But at the muscle level, again, the key concept is homeostasis. The protein, protein is really important in muscle, right? So maintaining the protein stability is a key thing to maintain exercise. And that goes down with age.
Right.
And that's why we think aging mice, even after a week of injection, we're already improving in their running. It's not because we're making new cartilage or muscle or whatnot. We were actually bringing back the lost communication in the body with age for many, many reasons. We're kind of bringing back that communication and activating their stress response.
Yeah.
And so maintaining their proteins better and their immune system to respond to it.
I mean, so I think it is fair to say that, the aged mice given MOTS-c just had more youthful cellular stress responses.
Yes.
Yeah. Wow.
More youthful homeostatic capacity.
Yeah. So, given the fact that people are using MOTS-c, there's an increasing number of people injecting themselves with MOTS-c. Say what we will about the rules and regulations, the people are doing it, and let's assume that we're talking about people who are getting genuine MOTS-c. What would you have people really think carefully about if they are going to use these things? One thing that comes to mind for me, based on what I've heard for you so far, is you certainly wouldn't want to use something like exogenous MOTS-c on a chronic basis.
Yeah, if I were to do it, I would definitely not do it chronically. But the effects are very different. So in mice, we do daily injections, and that's when we see the strongest effect. But it's a single dose. It's a single dose a day, and the half-life is pretty short of this synthetic peptide. It disappears, in a matter of minutes. And so it's not like we have a constant surge of cortisol the whole time.
Right.
It's almost like that one injection in animals was like a spike and then it disappears, which is what we want to do. So when we say chronic, I think the focus should be more on keeping that cycle going as any other stress does.
Yeah.
But it's hard to say, how do you determine the regimen? That's the clinical challenge.
Yeah. So you have to think about the timing of this on a couple levels. You could do one dose a day or multiple, and you were giving the mice one per day. You could also do many days in a row or a few days in a row, and probably both things are worth thinking about.
Yeah, and then when we shifted to the chronic lifelong treatment to see the effect on lifespan, we did 3 times a week.
Got it.
For many reasons. Again, the whole idea of the time becoming a biological parameter,
right?
A real biological variable. An intensive 2-week injection, we hypothesized was going to be different from an accumulation of month.
Yeah. And remind us, what was the basic result when you did the long-term 3 injections per week experiment?
It was pretty dramatic. We didn't extend lifespan because we extended health span of the mice, and lifespan studies take a lot of mice because as you go, mice sort of drop. And so we were not powered to do a lifespan study. We just shifted. But even with a small number of mice, we saw a continuation or maintenance of physical capacity with age. Of course, changes in the body composition. For example, older mice in their really later years, so we're talking about 30 months, and that's kind of a human equivalent of today as almost somewhere around 80. They
Yeah.
can't really run on the treadmill, even mice.
Yeah.
So they walk. So we put them on we made up this 1 minute walking test, for example, and most of the control mice cannot finish that 1 minute walking test at that
Yeah.
age. But almost every mouse that was left with MOTS-c completed that.
Wow. So they're not as old and frail looking, and they perform much better on these walking tests. How early in their lives did you start that 3 times per week injection for those
That was
elderly?
about 24 months of age. So
Middle age.
uh, around, so the way we see it is when you take a lifespan curve, the
Yeah.
moment when the lifespan starts to drop,
Yeah.
because it's almost like a waterfall for most species. If you compare that to humans, because it's hard to, correlate mouse age to human age, but
Yeah.
just using that stage where the mice start to drop and when humans start to drop,
Yeah.
it's right around that area. So we're talking about, for humans today, somewhere around 60s.
Yeah. So later middle age. Yeah.
Yeah, which
Um,
now is later in middle age because we live up to,
yeah.
almost 90 now.
Yeah, yeah. Okay, so you're improving. You may or may not have improved lifespan. You just weren't powered to see that, but you were definitely improving health span. They were
Yeah.
behaving in a more youthful way, much longer in their lives.
Right, right. And it's pretty amazing to see those 30-month mice on a treadmill still walking, which
Yeah,
is, and they could go more than a minute. We just stopped it because we called it a minute test.
yeah, yeah. Wow. Little time that we have left, I want to ask you. So MOTS-c is very fascinating. You've done a lot of great work there, and I'm sure there's more to come that we'll be able to talk about before too long here. The world of mitochondrial peptides in general. How many of these things do we know about? Do you think there's a lot more to be discovered? What do you think the landscape of mitochondrial peptides is going to start to look like?
So we have another one that we're preparing to publish soon. So for sure, there's one more. But Hasi's lab, Dr. Cohen's lab has discovered many other ones. Right now, there's about a dozen out there of these mitochondrial peptides. Some that we have discovered, my lab and Hasi's lab. But other people have found mitochondrial peptides as well. They're a little bit bigger than ours, but still, it falls into that category. But bioinformatically, there can be hundreds potentially.
Yeah, yeah.
Which really is crazy because that almost doubles our proteome if we start including these little peptides.
Yeah,
In
yeah.
the nucleus, there's a ton.
Right, right. Okay. Yeah, so still probably a lot more to be discovered. For those listening, so we just did a pretty deep dive on MOTS-c in this podcast. I'm actually talking to Dr. Cohen next week. So between those two, I'm trying to cover, almost everything that's out there to be known about MOTS-c so far. So if you like this episode, I would pair those together because we're gonna talk a lot more about MOTS-c next week. What is on the horizon for you? What are some experiments that you guys are doing today that you want to alert us to where you think we're going to learn some really interesting stuff about MOTS-c or other mitochondrial peptides in the near future?
Well, there's so many. I mean, my lab is almost like a collage of topics because, we basically just follow the data and it takes us to these really wild places. Aging is the ultimate goal for us. And mitochondrial communication is just an emerging field, and there's so many going on. It's almost more exciting than science fiction. George Lucas, who made Star Wars, if you're a Star Wars fan, you would know midichlorians. It's like the source of the Force. There's a scene. The story goes that George Lucas learned about mitochondria while he was making Star Wars, and he thought it was crazier than fiction. So he had to put it in a series, and they call it midichlorians. And it actually has Liam Neeson read from the textbook. And I think there was so much insight there. I don't think he understood this, but he somehow said it. In the movie, Liam Neeson sits down and kneels down and reads from the textbook saying, midichlorians are intelligent life forms that live within our cells. And these are words in the movie, in the cells. And we are symbionts with them. And he says something crazy. He says they continuously talk to us to provide the source of the force, basically. And when I heard that, I didn't really realize that. But going back now, the communication, the talking part, and the intelligent, form, I think that's where we really want to focus on.
Yeah.
Now, how much does the mitochondria how much they get involved in the computation of these environmental switches, right? And how do they process that? And how do they communicate that to the cell? That whole cycle, I think, is going to be really important to understand this. And it's not just going to be metabolic. We are focusing more on the immunometabolic interface right now.
Yeah. Are you generally, what's your general high-level take on the realm of peptides as a sort of new, I don't even want to say treatment modality, because people aren't necessarily using these only to treat problems. They're using these to increase their youthfulness or vitality or whatever you might want to say. Are you generally excited about the prospect of peptides as an emerging class of substances that human beings are going to use for health-related reasons? Or do you think we're getting a little overblown with the excitement on these things?
No, I think peptides have so much potential. I don't recommend taking MOTS-c right now just because we don't understand it fully. And there's always dangers associated when you enter unknown territory. But I think what's going on right now in this country is a reflection of that demand. People don't want to wait until they get sick to be prescribed the drug. I think the supplement field has demonstrated very clearly that there is an intention in a market to of preventative medicine, sort of a vaccination against aging kind of situation, where you want to do something to slow down the process. And I think these peptides are great because for many reasons, but like I said before, smaller peptides don't have a huge specificity. A lot of the drugs out there that are protein-based are very specific, and that's how FDA approves it. But the specificity comes with a lot of side effects because you are going to do something dramatic, go and clog some pathway very, very significantly. But like I said, these peptides typically won't have that level of specificity, but they'll be more involved in the general flavor of responses, which means it's more going to be in the optimization, fine-tuning of major processes rather than clogging or activating that process directly. And so therefore, I think from a longevity drug, preventative drug, I think peptides for that reason have a lot of potential. But of course, we need a lot of clinical trials to make sure we understand what we're doing. So do no harm is the first thing, right?
Yeah.
We have to cross that barrier first.
All right. Well, Professor David Lee, this was a great conversation. You've done some excellent work in this realm that I've been reading for the last week or so. So thank you very much for your time. And I look forward to seeing what comes out of your lab in the next, couple of years.
Thank you. Thank you. I really enjoyed our conversation too today.
Hey everyone, a great way to support my efforts is to visit my support page at mindandmatter.substack.com. A link to that page is in the episode description, or you can search for it on the Substack page itself. The support page has an up-to-date list of my affiliate partners. These are companies I work with, and if you buy those products through those links or using the discount codes provided, those will get you a great deal on a variety of products related to optimizing physical, metabolic, or mental health and vitality. These are all products I use myself, and some of them are directly related to podcast episodes or formulated by Mind and Matter guests. For example, there's a great product called Keto Citra, formulated by a kidney biologist who is on episode number 186. It contains the ketone body BHB together with several minerals in a precise ratio formulated with kidney health in mind. There are also links to products like physical technology devices to track your metabolic health, digital applications to help you find and learn about food and consumer products, and more. One product I use every day is the AquaTrue water filter. I recommend recommend this to everyone because it gets everything out of your drinking water. Heavy metals, microbes, endocrine disruptors, microplastics, you name it. Other charcoal or gravity-based filters that a lot of people use don't get everything out. They just get some of the stuff out. So I really like the AquaTrue product. You can look for a link to my affiliate partners on my support page or directly in the episode description to this episode, or you can go to mindandmatter.substack.com to find it there.